All right, well, let's look at translation. In a sense, you could use a good analogy for translation would be like a play in a theater that has 3 acts. Because translation, in effect, has 3 different phases or 3 different acts. And you know, the theater analogy is not such a bad thing. So we have Act 1. Now, if you're going out to a theater or something, what happens, the first act is usually where you introduce the main characters. They come in and give a little thing about the storyline. And Act 1, which is what we call initiation, is just like that. What we have to do during initiation is we have to bring in the main characters. That's the messenger RNA, that's certain proteins, that's the ribosomes, that's the tRNAs, all that type of stuff. So we have to get all the main characters in on the stage to set up the storyline. Now, in our play, you have Act 2, right? And that's often the main part of the storyline. Okay, like, you know, former Olympic champion murders his girlfriend in South Africa. Now that happened, so that's in the big news now. Okay, and Act 2 is when we're actually successively adding amino acids to this growing protein. And we call that elongation. Now, in the third part of our play, we reach climax and finally resolution of the whole thing. And finally, at the end, all the characters exit stage left or stage right, depending, right? Okay, well, our final act here, termination, where we end the storyline and end the protein synthesis. Termination. And eventually, all the characters exit. We disassemble the whole thing that was responsible for translation occurring. Okay, so we can look at it that. And also using the play analogy. You know, if you go up to a theater play or something, like, you know, out in New York City or whatever the case is, you go up there, okay, they're going to have all these different people who are the major actors and stuff, right? Okay, and here we have the major actors. The messenger RNA, the transfer RNAs, the ribosomes and stuff like that. But unless you look at the fine print on the playbill, there's a whole bunch of other people that are involved in that never show up on stage or show up real briefly. You never know they're there. And those are like the stagehands, the music people, the lighting people, the people that collect the tickets, the ad people, and all that kind of stuff. And without those folks, even though they may not be the major actors, without those folks, the show would not go on. You'd close up. Have you ever seen, you know, stayed in a theater long enough to see the credits at the end of the film? You know, there's like hundreds, if not thousands of people that are involved in various aspects of making a film. People that make the sets, make the guys that do the special effects explosions and all the computer graphics people that are making the aliens and the starships and all that kind of stuff. And, you know, so they get the little billing at the end. And without all those people, you would not have blockbuster films like The Hobbit or Avatar, stuff like that. Okay, well, same thing here. We have these unsung heroes of the whole thing that are called translation factors. And translation factors are proteins that come in. They're not part of the ribosome, but they come in, bind to the complex, do a specific task, and then leave. And without these translation factors, the show would not go on. They're absolutely essential for translation to occur. And we have initiation factors, elongation factors, and termination factors. And all of these proteins play critical roles in the process of translation, even though they are not part of the ribosome itself. Now, of course, the ribosome does plenty of stuff. So we have these types of things in our little 3-act play here of translation. All right, well, that's a little quick and dirty overview. We'll look at the gory details a little later on, but now let's look at a couple other aspects. First of all, let's look at what does the process of translation. Those are ribosomes. Each cell has scores, thousands, and you talk about oocytes, it can be billions and even trillions of ribosomes in the cytoplasm. And each ribosome is going to be able to be involved in translation. Now, ribosomes are complexes of RNA molecules and proteins. We have, in eukaryotes, prokaryotes are a little bit different, but in eukaryotes we have 4 ribosomal or rRNAs that make up the major portion of the mass and volume of these ribosomes. We also have in eukaryotes about 80 little small ribosomal proteins. They bind to the ribosomal RNAs. They tend to be highly basic, in other words, full of positive charge. They bind to the ribosomal RNAs into each other, and they do a number of things in protein synthesis. Now, ribosomes can be separated into 2 large portions, what we call the small subunit and the large subunit. Okay, the small subunit consists of about 30 or so proteins and a single large ribosomal RNA that we call the 18S. Now this term S is a reference to how fast you can spin it down in a centrifuge, and it's a crude indicator of size of something. The 18S ribosomal or rRNA. Now the large subunit, okay, it's got about 50 proteins in it that are different than ones in the small subunit, and it's got 3 of the 4 ribosomal RNAs. We've got something called the 5S, which is pretty small, something called the 5.8S, which is also pretty small, and the granddaddy of them all, the 28S ribosomal RNAs. Now, by the way, for those of you who've played around with prokaryotes and stuff in the past, the 18S is the eukaryotic equivalent of 16S. People who do gene sequencing and take like environmental samples and see what's in there and stuff, what they usually do is sequence the 16S rRNA to see what kind of microorganisms, prokaryotes are in there. The eukaryotic equivalent is the 18S, a little bit bigger. Now, in prokaryotes do not have the equivalent of a 5S RNA. Their 5.8S is a little smaller, and they call that the 5S in prokaryotes. Now, once again, that is not like the eukaryotic 5S. They don't have that at all. But they have a shrunken version of this one. And they have, the big RNA is the 23S in prokaryotes. Once again, very similar to the eukaryotic 28S, just we got extra stuff in there, extra sequences. Okay, and of course prokaryotic ribosomes are a little bit smaller. They have slightly fewer proteins, maybe about 20 and 35 or so. And the proteins are somewhat different. Now that's one reason why very important class of drugs that's widely used came into being. Because it turns out that prokaryotic and eukaryotic ribosomes, although there's a lot of similarities, and we have quaternary structures of both now. Just last year they got the quaternary structure of a eukaryotic ribosome. They had the bacterial ones for about past 5 years or so. But anyway, they got that, and they see a lot of similarities, but a few significant differences. But anyway, the cool thing about this, okay, well, certain organisms, specifically certain types of fungi, they are more into chemical warfare than Kim out in North Korea is, you know. They love chemical warfare. Because if you happen to be mold or something, you're competing with bacteria for the same environment. You're doing the same thing these guys are. Problem is, prokaryotes can divide a lot faster than you can. So, competing for the same environment, the prokaryotes have the edge. The bacteria have the edge simply because they can divide faster. They will overwhelm you. Well, when you're about to be overrun by a whole bunch of nasty fungi, what do? You break out the chemical weapons. And many fungi have produced chemicals that they secrete that affect specifically prokaryotes and don't affect themselves, or at least not unless the concentrations are way too high. Many of these things work by inhibiting prokaryotic ribosomes and not doing the same to their own ribosomes. We call them antibiotics. A lot of our first antibiotics, like streptomycin, penicillin, were directly isolate from fungal products. Now, you know, the drug companies chemically modify them to come up with new ones because, unfortunately, bacteria also evolve very quickly, develop resistance to all kinds of different antibiotics. But anyway, antibiotics are, for the most part, fungal products or derivatives of fungal products. And many of them work by specifically inhibiting prokaryotic ribosomes. So there are some differences here. But back to the eukaryote. Okay, now ordinarily the small and large subunit are separate in the cytoplasm. They float around and they only assemble together to make a full ribosome during the process of translation. So we'll see that later. Okay, a few basics of ribosomes. Now, one other kind of cool aspect about ribosomes is how are ribosomes themselves made? Their assembly pathway is rather interesting, and it involves shipping things in and out of the nucleus. So what we're going to do is we're going to make a couple nuclear pores here. Here, we'll put another one here. Okay, now how do we make ribosomes? Well, it's kind of like some of the manufacturing process. Sometimes you can buy something and it will say assembled in like China, made of components of US and Korean components. You can see that. And in a sense you could say the same thing about ribosomes. They are assembled in one location and components are made in two different locations and moved into the assembly plant. Okay, we'll start out here in the cytoplasm with a mature ribosome. Okay, here's a ribosome. Now, pre-existing ribosomes are going to make ribosome proteins for more ribosomes. So we have ribosomal proteins. Ribosomal proteins happen to have nuclear import sequences exposed on the surface. In other words, short sequences of amino acids that the nuclear import machinery are ran and the nuclear pores can recognize, and they get shipped into the nucleus. But not just anywhere in the nucleus. Inside the nucleus, there's a region of the nucleus. Now, it does not have a membrane around it. It's not a separate compartment, but it's a concentration of things. I'm going to represent that region by a dotted line to show there's no membrane here, and this is what we call the nucleolus. The nucleolus looks different from the rest of the nucleus because it's packed full of huge amounts of RNA. And you add RNA-specific stains. And we saw this when we looked at those frog oocytes. If you add RNA-specific stains, they stain the nucleolus much more intensely than they stain the rest of the nucleus. Because there's a lot more RNA there. And that's how it was first identified. Okay, the nucleolus is actually the assembly site for the ribosomal RNAs, or rather for the ribosomes themselves. Okay, so we're going to move in these ribosomal proteins into the nucleolus. Now, at the very center of the nucleolus, although you cannot see it, there happens to be a stretch of DNA from one of the chromosomes. And that stretch of DNA happens to have the major rRNA genes in it. Okay, we'll see a little bit more on that. That itself is a bit of a story. Now, these genes are transcribed. It turns out, because cells need so many ribosomes, we could not survive with just one copy of those rRNA genes. Instead, we have about 200, 250 copies lined up one after the other. There are spatial regions between the individual genes, but they're basically about 250 copies lined up in a row. And the reason why is we constantly have to produce large amounts of ribosomal RNAs, and you simply could not get that with one gene. So now we have multiple copies. Those genes are transcribed, and we have our own polymerase to do that. Polymerase 1 is what transcribes specifically the ribosomal RNA genes. So now we make ribosomal RNAs, which are right here. And under electron microscope, you can see this region is looking different from the outside region. So here's lots and lots of ribosomal RNAs. They, like messenger RNAs, will undergo certain processing, and we'll see that a little bit later. And they have their equivalent of a semi-spliceosome-like structure. It includes the SNRP called U3, which we don't find in the polymerase II based spliceosomes. So they have one of those small nuclear RNAs. Okay, well, now what happens here? We've got the proteins taken in, we've got the rRNAs produced, and now ribosomes have this magical property that we call self-assembly, which is really cool. What happens if you put the parts together? They spontaneously, without any catalyst, energy, or what have you, they spontaneously assemble to form large and small ribosomal subunits. Even though we're talking about extremely complex structures here. So we have this assembly. They automatically assemble themselves. And now we have large and small subunits.
And these are the ribosomal subunits we're talking about.
Yeah. Once these are completely assembled, they can get carried out through the nuclear pores and taken into the cytoplasm. Now the self-assembly thing is rather astonishing. Some viruses can, at least simple viruses, can do that. But think of this, you take a look at, say, the large subunit of the ribosome. You've got 3 separate ribosome RNAs and about 50 proteins. If you did an experiment, you can isolate ribosomes quite easily if you have the right equipment. So let's suppose you have this nice, it's a bluish, sticky, bluish, translucent kind of thing at the bottom of a centrifuge tube. So now I've got this stuff at the bottom of the centrifuge, a bunch of purified ribosomes. You take the ribosomes and using all kinds of biochemical techniques, let's break them apart and separate out each one of the RNAs and each one of the proteins. So now we've got about 60 separate test tubes. Each one has a ribosomal RNA or a ribosomal protein in it. We've completely broken it apart and separated into its components. Now, if you have this in the lab, and then somebody comes in that night and thinks, oh, where are all these test tubes? Just pours all this stuff into a single beaker, all these test tubes. You come in the next morning, you're ready to kill somebody. Okay. But you would find out remarkably that in that beaker, you would have fully assembled, fully functioning ribosomes. All you have to do is pour the ingredients together, and they assemble themselves. That actually was a nice way of understanding the function of ribosomal components, because what you do is have all your test tubes lined up, grab one test, well, it's got this protein in it, pour it down the sink, and then mix everything else together, and see if they assemble properly, see if they function properly, and what have you. So that was a way of molecular dissecting at the molecular level the function of ribosomes. What components did what. But this is an interesting kind of assembly pathway. Proteins are taken into the nucleus. Ribosome RNAs are synthesized in the nucleolus. They self-assemble, and then we take the fully assembled things out. So that's our basic assembly pathway of ribosomes. Okay, questions on that, folks? Okay, let's take a little look at the ribosome RNAs and how they're made. It turns out that the 28S, the 18S, and the 5.8S ribosome RNAs are coded for by a single gene. And there was about 250 copies of that gene in this cluster of RNA genes. So 28S plus 18S plus 5.8, single gene. So here, let's say we have a gene here, and there will be another gene later on. Of course, we've got all the promoters and this kind of stuff. There are polymerase I specific promoters, and they have their own version of a basic complex. By the way, our TATA binding protein is also involved in the polymerase I promoter and plays a similar role as the cornerstone of that. So we have our promoters, which is I'll put P here of each one of them. Okay, now.
Dr. Sauterer, so
yes.
This right here is assembly of...
This is the genes.
Okay.
Okay, so this is DNA here. Now we're going to transcribe one of these guys into nuclear RNA, our precursor. We have a single, I'm just going to enlarge it, a single gene, a single giant RNA. We often call this the 45S RNA because it's gigantic. It's huge. Now along come few little spliceosome-like complexes, including the U3 and a couple of these other SNRPs, the RNA protein complexes that make up spliceosomes. And we slice and dice and cut it into 3 portions. We have one portion is the 18S, and then some parts are discarded, in fact, almost kind of like what we have in the case of introns and exons. We have our second thing, which forms the 28S, another discarded region, and finally our 5.8S. Okay, so in other words, each one of these genes codes for 3 of the 4 ribosomal RNAs. And there we've got it. Now the question is, what about that last ribosomal RNA, the 5S? Okay, turns out the 5S genes, and there are a couple hundred copies of those in a cluster, are located on entirely different chromosomes. They're not found in nucleolus. They are transcribed, and they're transcribed quick because they're small genes. And then those RNAs, those precursors of the 5S RNAs, are then shipped in nucleolus for final processing. And now we've got all the RNAs we need, and we're ready to rock and roll. So here we see with the polymerase 1 based genes, we're seeing transcription, their own type of basal complex, their own polymerase, their own dedicated polymerase, and their own specific type of RNA processing, which is in this case mostly cutting stuff off and cutting out sections that you don't want. So that's what we see for the formation of the ribosomal RNAs. Okay, questions on that?
So this is how all the 18, 28, and 58S all come to be? Is that what this is?
Yeah, yeah. They're cut from a giant precursor RNA, which is cut into 3 pieces. One's the 18, one's the 28, one's the 5.8. And then whatever else is just tossed in the garbage.
Okay, thank you.
Okay, all right. Okay, let me just...
That's probably a good time too.